Hello everyone, in this video I am going to discuss about neurulation, and it is nothing but the process of formation of neural tube which will become brain and spinal cord, and it occurs only in case of vertebrate embryo after 3 weeks of fertilization. Now let me introduce you with a new word, neurula, and it's nothing but the embryo where neurulation process is going on. It's known as neurula. Now come to the origin of neurulation, and it is ectodermal in origin. Now see what happens after fertilization. We get a diploid zygote, and this zygote become morula, blastula and gastrula due to repeated mitotic division. And during this gastrula stage, we get 3 layers of cells: ectoderm, mesoderm and endoderm. And this ectoderm is the origin of neurulation. That means neurulation process begins from this gastrula layer. And what happens in this ectoderm layer? The dorsal mesoderm signaled dorsal ectoderm to specify some of its portion known as neural ectoderm. And this neural ectoderm become epidermis, neural crest and neural tube. This neural ectoderm comprises of 3 parts: epidermis, neural crest and neural tube. And this epidermis later form skin, hair, nails, anterior pituitary. And this neural crest form PNS, adrenal medulla and Schwann cells, etc. And this neural tube form brain, most important, retina, spinal cord, motor neuron, neural pituitary, etc. Now come to the types of neurulation, and it is of 2 types. Number 1 is primary neurulation, and in this primary neurulation what happens? Formation of neural crest, neural tube and epidermis occurs. And in this secondary neurulation what happens? Differentiation and commitment occurs in the cell. That means after secondary neurulation, the cell, that means neurula, is going to form neurons and the part of neural systems, etc. This primary neurulation comprises of 4 steps: shaping and folding, elevation, convergence and closure. Now come to the shaping, and in this shaping step what happens? The neural ectoderm divides into 3 sets of cells, and these are neural plate, neural crest and neural epidermis. And this neural plate is the thickest and the elongated one in comparison to neural crest and epidermis. Now see what happens in folding step. In dorsal mesoderm, right below to the neural ectoderm, we see notochord, and this notochord stimulates in this MHP. MHP means median hinge point of the neural plate to decrease its height, and this leads to the formation of a groove in the neural plate, and this is known as folding. Now come to the second step of primary neurulation, that is elevation. And in this elevation what happens? See, after stimulation of notochord, MHP cell decreases its height and forms a groove, and after that these 2 epidermis layer apply a motive force on this neural plate and forms 2 neural folds here, and due to these neural folds the groove of MHP become bigger. Now come to the third step of this primary neurulation, that is convergence. And what happens here? See, due to repeated force of epidermis on these neural folds, the groove become bigger and arranged in such a way that the epidermis and this neural plate comes to join each other, and here form another hinge point known as DLHP. This is dorsolateral hinge point. It is just like MHP, and this is convergence. In the convergence step, we see that the neural plate and epidermis come to join with each other, and in this closure step what happens? See, a protein named as N-cadherin comes, and this protein separate this epidermis and the neural plate, forms epidermis in outer portion neural crest in middle portion, and at last neural tube in the inner portion, and this is known as closure. And after that we get a epidermis layer, we get a crest, that is neural crest, and we get a proper neural tube. This is known as closure. Now come to the factors affecting this neuralization process. Number 1, gene. There are some genes like PAX, SHS. This is sonic hinge hop. And number 3 is open brain. And these are responsible for this neuralization process. If there is a mutation or some defect in these three genes, the neuralization process will be defected. Second factor is dietary product, and there are two dietary products are responsible for this neuralization process. Number 1 is folic acid, also known as vitamin B12. Number 2 is cholesterol. And if these two dietary product is absent in a pregnant mother, what happens in the embryo, developing embryo, the neuralization process will be defected. Now come to the secondary neuralization. And in this secondary neuralization, what happens here? Two axes are formed. Number 1 is anterior posterior axis, and number 2 is dorsal ventral axis. And these two axes are responsible for the formation of neural system and the neurons. Now come to the anterior posterior axis. And what happens here, see, due to the force of neural fluid of neural tube on the neural wall, in the anterior portion, 3 chambers are formed or vesicles are formed, and these are known as, number 1 is prosencephalon, and it becomes forebrain. The second one is mesencephalon, and it becomes midbrain. And number 3 is rhombencephalon, and this becomes hindbrain. Later, this prosencephalon divided into two parts, telencephalon and diencephalon. This telencephalon becomes cerebral hemisphere, and this diencephalon become hypothalamic and thalamic region. Mesencephalon remain undivided, and rhombencephalon divided into two parts, metencephalon and myelencephalon. And this metencephalon becomes pons and cerebellum, and this myelencephalon becomes medulla oblongata. That means from this anterior posterior axis, brain parts are formed. Now come to the dorsal ventral axis of secondary neuralization. And what happens here, see, this notochord started secreting HHH protein, this HHH protein on the ventral side of the neural tube, and this formed floor plate here. This is known as floor plate. After that, from the epidermis, BMP4 and BMP7 proteins secretes, and this BMP4 and BMP7 acts on the dorsal part of the neural tube and forms roof plate. Now see, this BMP4 protein also trigger the formation of TGI family proteins. Then these are BMP4 itself, BMP7, activin and dorsal. And these TGI family proteins forms a gradient, protein gradient here. And from dorsal to ventral part, the concentration of this protein become lower. Now see, this TGI family protein gradient started forming neurons in neural tube, and at the very first, sensory neurons are formed on the dorsal part, because on the dorsal part the TGI family protein gradient is very high, and after that interneurons are formed, and at last motor neurons are formed. And we see that in secondary neuralization, neurons are formed, and in the primary neuralization, what we have seen, the brain parts are formed, and thus the neuralization process occurs. So I hope you all understand this topic. So thank you all.